One of the biggest misconceptions of evolution is that individuals evolve. A beaver can't mutate itself into being stronger or faster, and mutations are no guarantee of a perfect solution to a problem. Selection can only act on existing variation and so is limited by historical constraints. Populations change over time because those with a low fitness die before they can pass on their genes. The shift in phenotypes due to that selection is really what drives evolution. Populations are groups of individuals in the same location that have the opportunity to mate with one another. For example, caribou in Alaska and Yukon are split into two populations, the northern porcupine herd and the southern 40-mile herd. The populations overlap, which means they can mate, but they don't. Why not? It is partially because these animals are nomadic and don't end up in the same place at the same time. The other factor is that they prefer to mate with their own kind. They have been separated for long enough that the other population appears strange to them. In other words, mating is not random. The modern synthesis was driven by improvements in our understanding of genetics. There was a switch in thinking about traits evolving to thinking about the underlying cause of traits evolving, genes. What do I mean by genes evolving? Instead of thinking about phenotypes, like green frogs and red frogs and purple frogs in a population, think about their gene pool, which consists of alleles in different frequencies. See how the data is completely different. There's a lot of polymorphism or phenotypic variation that is external and easy to see, but what is going on in the DNA? Recall that Darwin had no idea about DNA or mutations as the source of new phenotypes. But really, mutations are not as important for generating new combinations of phenotypes as recombination, also known as crossing over. Meiosis, the form of cell division necessary for sexual reproduction, is what creates phenotypic variation. This matters because larger populations will have a larger probability of having both rare mutations and more combinations of alleles from different genes, making larger populations more genetically diverse, but also these alleles are more likely to be lost in the crowd. Sometimes we observe gradual changes in a phenotype over a landscape. For example, yarrow plants go from tall at low elevations to short at high elevations. This is due to a combination of selection in different directions at different elevations and constant gene flow in the middle, which stops the formation of separate populations. This is referred to as a cline. How would you describe the population of this fish? These are examples of disruptive selection, where traits on the extreme are ideal in these environments. Another form is directional selection, in which a single extreme is preferred. Lastly, stabilizing selection can favor individuals in the middle of the distribution of traits. Let's say you have a population of narwhals that vary in tooth length. If males with the largest tooth are favored by females, what mode of selection would they be expected to be under? The mechanisms that I've been describing, selection, non-random mating, mutations, gene flow, and small populations evolving quickly, are all related to one another. They are mechanisms for evolutionary change. If they are not occurring, the allele frequencies within a population should be stable over time. This is called Hardy-Weinberg equilibrium. It is a model for the conditions that would cause evolution to not occur. It should rarely ever happen in nature that all of these conditions are met, but it is a helpful framework to understanding evolution. Let's let the Amoeba sisters go over the math.
Do you like math? Sometimes when people are starting out in biology, for some reason they assume there is no math in biology, but then they soon find out they were wrong. My math teachers were telling the truth when they said math isn't everything. Chi-squares, osmotic pressure calculations, standard curves with gel electrophoresis, Punnett square ratios, these all have math. But one of my favorite topics that involves biology and math and is also absolutely fascinating is the Hardy-Weinberg equilibrium. It got its name from a mathematician and a physician, and it states that a population's allele and genotype frequencies are constant, unless there is some type of evolutionary force acting upon them. A reminder from our ecology videos, a population in this case is a group of organisms that are all the same species and can breed with each other and have fertile offspring. However, there can be variety among a population as they're not clones of each other, but based on Hardy-Weinberg equilibrium, the population's allele and genotype frequencies would remain constant if no evolutionary force acts upon them. If we have these frogs here, assume they are all the same species in this example, but there are some slight differences. In our simplified example, some of the frogs are lighter green and some are darker green. We're going to use a simple genotype in this example. All frogs in this population will be either big G, big G, big G, little g, or little g, little g. There's also an allele frequency in our population. We're going to say here that 60%, a frequency of 0.6, of the alleles are big G. 40%, or a frequency of 0.4, of the alleles are little g. Notice the percentages add up to 100%, and the frequencies add up to 1. Now, to be in Hardy-Weinberg equilibrium, we have to have 5 assumptions. Number 1, no selection. No natural selection is acting upon these frogs. That means neither dark green nor light green will have any impact on reproductive fitness. Number 2, no mutation. Baby frogs inherit genes from their parents and there are never mutations. Number 3, no migration. Frogs can't come in, frogs can't go out. Number 4, large population. There are a lot of frogs. It turns out small populations are more vulnerable to genetic drift, by the way. See our genetic drift video. Number 5, random mating. The frogs mate without any specific choice. All of these 5 assumptions must be kept in order for Hardy-Weinberg equilibrium to happen. So, in real life, does this generally happen? No. For example, in real life, maybe in a certain environment, predators can more easily see the light green frogs, so perhaps they are eaten more and have less reproductive fitness. So if Hardy-Weinberg equilibrium is unrealistic in nature, why does the Hardy-Weinberg equilibrium matter? That's where the math comes in. The Hardy-Weinberg equilibrium gives you this baseline to compare how an evolving population could compare to one that remains constant without evolutionary forces acting upon it. And now let's explore the math. So there are 2 equations in Hardy-Weinberg equilibrium that we will focus on. We're going to start with the first one. P plus Q equals 1. In this equation, P is the dominant allele frequency in the population, and Q is the recessive allele frequency in the population. By the way, Hardy-Weinberg equilibrium doesn't mean that P has to equal Q, and the dominant allele frequency in any population doesn't have to be larger than the recessive allele frequency in a population. That's a misconception because dominant alleles aren't always the more common allele. The equation does say that the dominant allele frequency and the recessive allele frequency have to equal 1. So, in my example here, if I say 60% of the alleles are big G, 40% of the alleles are little g, 0.6 is P, and 0.4 is Q. So this equation, P plus Q equals 1, is for those allele frequencies. But what if I wanted to know the genotype frequencies? Meaning, I wanted to know the frequency of frogs that are homozygous dominant, which is big G, heterozygous, which is big G, little g, or homozygous recessive, which is little g. Then I can use this other Hardy-Weinberg equilibrium equation, P squared plus 2PQ plus Q squared is equal to 1. So P squared is the homozygous dominant frequency, the frequency of big G in this case. 2PQ is the heterozygous frequency, so big G frequency in this case. Q squared is the homozygous recessive frequency, so little g frequency in this case. Let's plug those previous P and Q values in. P squared would then be 0.36. 2PQ would be 0.48. Q squared equals 0.16. Cool, huh? The only thing is that when calculating Hardy-Weinberg equilibrium problems, you don't always have the P and Q values. So we're going to do an example. New population of frogs, so forget the previous frequencies. This is a new frog population in a new frog land, but we'll still use the same allele letters. So here's the information you get for this new population. There are 500 frogs, and of those, 375 frogs are dark green. The rest are light green. With that information, please solve all genotype frequencies and all allele frequencies if in Hardy-Weinberg equilibrium. Step 1. Determine first whether you're going to work with the first equation or the second equation. Since I'm working with individuals here that have genotypes, I'm going to work with the second equation first. Step 2. Figure out what value you can determine. So 375 frogs are dark green out of 500 total. That would mean 125 frogs are light green since there are 500 frogs total. But I can't use whole numbers like that, as both of these equations ultimately are equal to 1. I need frequencies for these equations. Now, I could say 375 dark green frogs out of 500 total frogs is 0.75, but all I know is that would be the frequency of dark green frogs. The problem is dark green frogs could have genotype big G, or they could have big G, little g. I can't assume they're one genotype or the other, so I shouldn't use that value. The recessive genotype, resulting in a light green trait, is safe to use though, because I know that light green frogs are the genotype little g. They can't be anything else. So 125 frogs out of 500 frogs equals 0.25. That means, from the equation, the value q squared equals 0.25. Step 3. Take a value you solved from the previous step and calculate from there. So if I know that q squared is 0.25, then I could go ahead and solve for q. If q squared is 0.25, I can determine q if I take the square root of 0.25. Therefore, q is equal to 0.5. That's the allele frequency for the recessive allele little g. If I know the q value, I can find out the p value using the first equation. Since p plus q is equal to 1, now I know that p is equal to 0.5. That's the allele frequency for the dominant allele, big G. Now I have p and I have q, and I can take care of everything else. I can use the second equation to determine the homozygous dominant frequency, the heterozygous frequency, and I already knew the homozygous recessive frequency from the beginning. Plugging those in, p squared equals 0.25 for the genotype frequency big G, 2pq equals 0.5 for the genotype frequency big G, q squared equals 0.25 for the genotype frequency little g. Phew, that's a lot.
So, a population at Hardy-Weinberg equilibrium is not evolving as evidenced by allele frequencies staying the same. All 5 conditions must be met for this to be true. If you imagine a population of flowers, generation 3 on the diagram, that are mating randomly, the percent of genotypes and phenotypes won't change in future generations. P and Q in the equation are the relative frequencies of each allele. In this model, there are only 2 alleles, but the formula can be modified to include more. P squared and Q squared represent the frequencies of the homozygous genotypes, and 2pq represents the frequency of the heterozygous genotype. P and Q are fractions between 0 and 1. They also can be added together and must add up to 1. Let's try an example. A population of sheep is in Hardy-Weinberg equilibrium. The allele for white wool, big W, has an allele frequency of 0.19, and the allele for black wool, small w, has an allele frequency of 0.81. What is the percentage of heterozygous individuals in the population? You were given both P and Q. What you were looking for was 2pq. Let's try another example. Cystic fibrosis is a recessive condition that affects about 1 in 2,500 babies in the Caucasian population of the United States. What is the percentage of carriers in the population? Most evolutionary change is created through 3 forces. These forces stop Hardy-Weinberg equilibrium from occurring. The first is selection. For example, a predator may eat all the blue caterpillars. The second is genetic drift. Imagine a honeybee pollinates certain flower types at a higher frequency one year, but the bee doesn't actually care what flower it goes to. The flower population will randomly drift. Make sense? Perhaps a little more from the Amoeba Sisters.
So here's a question for you. Have you ever compared the candy you get on Halloween with your friends? My sister and I, a bit competitive in the sport of Halloween candy hunting, we both had preferences of which kind of candy we hoped to get in our buckets each year. And as for me, I love gummy bears, and every once in a while someone would have them mixed in a big bucket, and I remember watching with intense focus the hand that picked out the candy hoping for gummy bears. Getting those gummy bears was always total luck, total chance. Well, unless of course the entire bucket they had was actually gummy bears, but that would not be luck. No, that would be pure bliss. But that's never happened to me, yet. So why bring this up? Well, what if instead of candy we were talking about insects? And they weren't in a Halloween bucket because that'd be kind of gross. They're on the sidewalk. And when you're scooter riding through the neighborhood on Halloween to collect candy because scooters may or may not be our super awesome childhood mode of transportation, you squash some. Accidentally, of course, because you like insects overall. Were the ones that survived better adapted? Did they have some special ability to foresee events or to move your scooter away from them? Well, assuming they were all just there on the sidewalk in the path of your trick-or-treating, then no. In this random event example, the survivors were not better adapted. They were lucky. It was chance. And the ones that were squashed were, in this case, at the wrong place at the wrong time. And that's what genetic drift is. Genetic drift can change allele frequencies in a population. Remember, alleles are a form of a gene, but genetic drift is random, unlike natural selection. Recall that in our natural selection video, organisms that have traits that result in high fitness are able to pass those alleles on to their offspring. This is where that survival of the fittest comes into play, but not genetic drift. With genetic drift, the organisms aren't necessarily more fit. They just have won the game of chance. So let's look at a representation of genetic drift known as the bottleneck effect. I always like to remember this one by focusing on the word bottle. Well, let's say that we have a bottle here and we fill it with our Halloween candy. If we shake it out and we only get a few of the candy pieces, does it necessarily represent the whole bottle as far as the candy frequencies you got out? Nope. In fact, one of the types of candy pieces isn't even there. That type is totally eliminated. We'll consider a natural disaster, such as a forest fire. The surviving organisms weren't better adapted. They were likely just in an area where they weren't directly affected. But the survivors don't happen to represent the original population, so there's definitely going to be a new allele frequency among the surviving population. Bottleneck effect. Another time to see genetic drift is when you're talking about the founder effect. In this case, you may have organisms that have founded an island or some new area. The few organisms that arrive to start a new population do not necessarily represent the original population that they came from. Let's take a case where maybe some seeds that are dispersed by wind end up in a new area that happened to be one that was ideal for their growth. This new population grows. Well, those seeds that landed in this new area may not necessarily represent the original population of plants from which they came from. It's a random sample, founder effect. In summary, genetic drift can make some big changes to allele frequencies, and it's a mechanism for evolution. So one last thing. Which type of population do you think would be more vulnerable to genetic drift? A big population or a small population? If someone is just doing some intense candy hunting and by accident scooter rides over a few insects, look at the results in the small population versus the big population. As you can see, this random event has the potential to cause more change from the original in the small population. See, there weren't as many representatives there in the first place. So in many incidences of genetic drift in populations, the impact on small populations is especially significant.
Lastly, gene flow can mess up Hardy-Weinberg by introducing genes from a different population. A less common problem is a genetic bottleneck, where the population size drops drastically in a short amount of time. Random alleles are lost, the allele frequency changes, and genetic diversity drops. You might think that genetic diversity would easily be lost because some traits are always favored over others, but there are also mechanisms that maintain genetic variation. For example, the simple fact that we have 2 copies of each gene, called diploidy, means that we carry hidden alleles by being heterozygous. Another mechanism is balancing selection, where the most fit changes over time. For example, the pepper moth maintains light and dark morphs in its population because the environment varies in both space and time based on pollution levels, predators, and the tree bark color. Another form of balancing selection is heterozygote advantage. A person with the heterozygote condition for sickle cell anemia is both resistant to malaria and only gets a mild form of sickle cell disease. Thus, the homozygous forms are maintained in the population. And remember that most traits are not under any selection, or at least any strong selection. Selection can sometimes be frequency dependent. In the case of happy-faced spiders, having more variation in coloration confuses predators and therefore benefits the entire population. This is referred to as group selection. Lastly, sexual selection is an engine for speciation and runaway evolution because often traits that are unusual are favored. I studied jumping spiders for my master's degree, and the females pick males that do the best job of getting their attention. Here are just 2 examples.
Male peacock spiders have tiny, beautifully patterned butts. These are used to create a series of elaborate signals during their mating dance. However, this is not just some flossing at a middle school prom which was held in the gymnasium because someone got the measles. Rejection of the male's advances can leave him much less alive. Dead, really. To understand the complexity of the courtship, we will examine 2 different species of Moratus. Hey, I'm Pete.
Hey, I like your little beard. It's cute.
Thanks. I can do this.
Is that yoga?
I think so. It hurts. Oh, check this out. It goes up.
That's so pretty.
And if I move it,
You look like a half-eaten butterfly.
right?
Is that cologne?
Yeah. Well, it's body spray. You want to smell it?
Okay.
I put it on all of my pits, but I think these 2 are like the easiest for you to.
Oh, I like it. It's.
Oh, sorry.
No.
Sorry.
It
Too
was
close.
nice.
Sorry. Yep.
Hey, I was on my way to Under a Log. If you wanted to.
No, yeah, totally. Under a Log is like my favorite place ever.
Cool.
Ooh. Hi, I'm Pete.
Cool, Pete. Don't give a f. Get over here. Now.
All right.
Okay, turn it around. Let me see what you got.
So what's your-
Don't talk.
Okay.
Boo.
What are you doing?
Do you know the game Marry Kill?
Yeah.
Well, I'm gonna play that with you.
All right.
But I don't want to get married, okay?
Is it weird that I'm kind of turned on right now?
Pete, shake that ass for me.
Like right now?
Now.
Let's do it.
Look at this butt. I know you can't help it. Waiting in line, see your butt like mine, you feel helpless. This butt don't sweat, but it can knit you a sweater. You ain't never gonna see a better sweater, weather sweater than a button-knit sweater. Happy time. I know you see this, honey. It's such a shiny honey.
That was good, right? I feel like energized. Why are you smiling? What?
Get your ass over here.
Just so you know, I don't really know how this works. Are you sure this is right? Because it doesn't seem like it's gonna work geometrically.